Now we will be discussing about the glycosaminoglycans. What are the glycosaminoglycans? They are unbranched long heteropolysaccharides, amino and acidic sugars. So they are the examples. Let's see. Chondroitin sulfate. This is present in the bone, cartilage and aorta. Keratin sulfate, which will be present in the cornea and the connective tissue. Dermatan sulfate is present in the dermis, arteries, valves like heart valves and also sclera. Heparin sulfate is most important for presenting in the basement membrane of the kidney. Why it is important? Because it contains of the negative charge so that it can repel the whichever the positive charges. Next, synaptic vesicles of the neurotransmitters and endothelium. Heparin sulfate. Heparin sulfate is different. Heparin sulfate is the anticoagulant and also we are going to use for the LPL measurement. Hyaluronic acid present in the vitreous body. Next, coming to the mucopolysaccharidosis. These are the diseases if they are deficient with this glycosaminoglycans. Let's see, the most important, the most common overall is the Sanfilippo disease. In the Sanfilippo disease, what happens? This dermatan sulfate is deficient, so that the patient will be having the deficiency of these conditions and also mental retardation. Next, other two are very important for the examination, Hunter and the Hurler. Hunter, remember this way, Hunter is searching for his eggs with his eyes. Okay, this is the mnemonic. Why? Because it is the only X-linked recessive of the mucopolysaccharides and he does not have the eye defect. I told that he is looking with his eyes. That means no eye defect. So, and coming to the enzyme that is deficient is the alpha L iduronate sulfatase is the deficient for this patient. Next, Hurler syndrome. It is L iduronidase, which is a deficiency enzyme, and mental retardation, eye defect will be seen like this corneal clouding. This eye defect will be present in this patient. What are the common symptoms in all mucopolysaccharide patients? Umbilical hernia, corneal clouding, coarse facial appearances, claw hand. These are the symptoms that we are going to see in these patients. Next, moving on to the glucose metabolism. In the glucose metabolism, first understand what is the basic concept. Whenever you ate the food, after immediately you ate the food, your blood levels will be increased with the glucose. So this glucose will be increasing the insulin glucagon ratio so that you will be breaking down the glucose into the glycolysis and excess of the glucose will be converted as a glycogen. That means glycogenesis will be seen in the liver. Okay, this is the when you ate. When you are fasting, what happens? There is no food at all. What happens? Low glucose and low glucose will be activated by the phosphorylation process. So that means insulin glucagon ratio, whichever is decreased, it is going to convert this glycogen we have converted, right? Excess glycogen that will be breakdown for the glucose energy. And after that, whichever the non-glucose products, they also going to helpful to increase the glucose level to compensate this low glucose during the fasting state. How that it works? So immediately when you ate, glycolysis for the ATP production. For example, let's say one whole night you didn't eat anything. So morning you are going to break down the glycogen, glycogenolysis. After the 24 to 48 hours, what happens? Gluconeogenesis. Non-glucose products is going to convert the glucose. Next, 48 hours to 5 days. That means your body is going to use the ketone bodies for the energy production. And more than 5 days, muscles will break down and that will cause the proteolysis. This is how the pattern whenever you out of food or you are in the hunger, starvation or the fasting stages. Next, glycolysis pathway. In the glycolysis pathway, whenever you had taken the food and immediately after that what happens? The glucose is going to converted as a irreversibly glucose 6 phosphate. Okay, irreversible. Hexokinase is going to convert it and it will consume 1 ATP. And these glucose 6 phosphate is converted into the fructose 6 phosphate and then fructose 6 phosphate irreversibly converted as a fructose 1,6 bisphosphate. Okay, this is the complete, this important step is very important because this is the rate limiting step. And overall this glyceraldehyde is going to converted as a 1,3-BPG, okay, biphosphate glycerate and 3-phosphoglycerate and phosphoenol pyruvate and pyruvate will be converted. So this cycle will be vicious until the energy is produced. So I don't want you guys to remember the entire cycle of the glycolysis, whereas the TCA cycle. But what are the points that you have to take note? Here are those important topics. For example, where is the site that the glycolysis is occurring in the cytoplasm? And what is the rate limiting enzyme? As I said, the fructose, when it is fructose 6-phosphate converted as a fructose 1,6-bisphosphate, this phosphofructokinase is going to be the rate limiting step. And what is the irreversible reactions? Hexokinase, phosphofructokinase, and fructose kinase. Okay, these are the important for the irreversible equations. Next, net ATP production. So basically, we are going to produce the 9 ATP. How? Let's see. So here 1 ATP is consumed for the glucose conversion and 1 ATP for the fructose conversion. Okay, 2 ATPs are already used. Next, we produce when we are converting the glucose glyceraldehyde to the bisphosphoglycerate. How many we produce? 2 NADH. That means 1 NADH is equal to 1 NADH is equal to 2.5. That means we have produced 5. Okay, and another ATP here 6 and another ATP is 7, right? 7 ATP we produced and here we already consumed. So total 9 ATP out of minus 2. Okay, 9 minus 2, that means we have produced a total of the 7 ATP. If they ask net ATP, please prefer the 7 ATP that we have produced. Next, pyruvate can be converted as a lactate. This is an anaerobic cycle. So in the anaerobic cycle, we are going to consume the NAD. And what happens? So 7, you were supposed to produce the 7, but during the anaerobic cycle, you are going to produce only 2 ATP. Okay, anaerobic lactate cycle 2 ATP, aerobic cycle pyruvate, then 7 ATP. Next, pyruvate is going to connect to the TCA cycle. How? That is with the help of the PDH enzyme. What is this PDH enzyme? Pyruvate dehydrogenase. It is going to use the multiple vitamins. Example, B1, B2, B3 and B5 along with the lipomamide. So PDH complex is very important. It is going to use the 4 vitamins plus the lipomamide to connect with the TCA cycle. Another important thing that you have to remember. Next, inhibitors. Fluoride oxalate. It is going to inhibit the anolase. Here you can see this anolase should be inhibited. Why? Whenever you take the gray vacuitainer, you are going to connect with the glucose. You have to see the glucose levels. But glucose is going to convert to the pyruvate because it has to break down to produce the ATP. So to prevent this breaking down, you have to inhibit this anolase so that you can estimate the glucose. Okay. So with the help of the fluoride oxalate in the gray vacuitainer and iodoacetate, arsenate, these are the poisons that is going to inhibit the glyceryl 3 PDH. Here you are going to inhibit, entire cycle will be stopped. Next, coming to the TCA cycle. In the TCA cycle, don't have to remember the entire pathway, but know the important points. Where is the site? Mitochondria. How much net ATP? 10 ATP we are going to produce. And this is the amphibolic pathway. Why we have told that this is the amphibolic pathway? It is important for the carbs. Okay, carbohydrate metabolism. See here the pyruvate TCA cycle. Carbohydrates, yes. Next, protein urea cycle you are going to see where these fumarate and also citrate, which will be helpful for the urea cycle. Fats, fat metabolism, oxalosuccinate, oxaloacetate, these are all going to be helpful for the fat metabolism also. So this is going to give us the all 3 important pathways that we can get from here. Next, irreversible pathways. Here you can see only the citrate synthase and also here the alpha ketoglutarate is the irreversible pathways. Next, vitamins that are used, vitamin B5, B1, B2, B3 are used in the cycle. Inhibitors, fluoroacetate, this is going to block this aconitase, and next arsenate is going to block the keto acid dehydrogenase, and malonate can block the SDH here. Okay, these are the points that you have to remember from the glycolysis and the TCA cycle. Next, glycogenesis. In the glycogenesis, whenever there is a excess glucose, it is going to convert it as a glycogen. Where it is happening? It is happening in the cytoplasm after you eating in the liver cycle. And rate limiting enzyme is the glycogen synthase. What is the branching enzyme? You have to connect the glucose, right? Glucose plus glucose plus glucose. If you want to connect this glucose, you want to make the glycogen. right? For this glycogen, we need the branches. For this branching only, we are going to use this branching enzyme. What is this branching enzyme? Alpha 1,4 alpha 1,6 glucone transferase is the branching enzyme to make the glycogen. Next, glycogenolysis. Glycogen is breaking down into the glucose. So for that, the rate limiting enzyme is the glycogen phosphorylase. When this is activated, when you are in the fasting state, and where this is occurring, same in the cytoplasm. Only the difference is it is in the fed state, it is in the fasting state, and liver plus muscle will be involved here. And glycogen is breaking down back into the glucose. That means whatever the branches that you are created, you have to break that. So that's why we have that debranching enzyme. Alpha 1,4 and alpha 1,6 glucosidase is going to be our debranching enzyme. Next, gluconeogenesis. What we have told, this is also during the fasting stage or the starvation more than 48 hours. What happens? Non-glucose products. For example, alanine is an amino acid. It will going to be the Cahill cycle. Lactate, it is an anaerobic cycle going into the Cori cycle. And glycerol, this is a fatty acid. Next, valine, methionine, isoleucine, threonine, odd chain fatty acid. All these are going to helpful to produce the glucose. End product, they want to create the glucose for our ATP production. So this is the gluconeogenesis. From non-glucose products, we are going to convert into the glucose. Okay, where we added the amino acids, fats, anaerobic cycles, all we have added. Where this is occurring? This is occurring in the both cytoplasm and the mitochondria. Next, galactose metabolism, you don't have to remember much. Galactose is converted into the galactose 1-phosphate and then converted as a glucose 1-phosphate with the help of the galactokinase and the Gal1 gene. Next, fructose also same that way. Fructose will be converted as a fructose 1-phosphate with the help of the fructose kinase, and with the help of the aldolase, it is converted as a aldehyde substances. Next, HMP shunt, glucose will be converted as a glucose 6-phosphate, and glucose 6-phosphate which will be converted with the help of the glucose 6 pyruvate dehydrogenase. It is converted as a phosphoglycerate and ribulose 5-phosphate, which will be helpful for the DNA RNA synthesis. Okay, and end product that is produced by this is the NADPH, which will be helpful for our the cycle that are catalase or the reductase, which will be helpful for our oxidation processes. Next, diseases that you have to remember in this chapter. The glycogen storage disorders. Remember, 1A is von Gierke disease. This is deficiency of the glucose 6-phosphate. Next, Pompey's disease type 2, acid maltase is the deficiency enzyme. Cory's debranching enzyme. C for Cory's, D, C, D. A, B, Anderson and the branching enzyme. M for M, McArdle's and the muscle phosphorylase. Hers, H for H, H for hepatic phosphorylase. Next, by the features, overall the most common is a von Gierke, severe hepatomegaly, lactic acidosis, hepatomegaly, hyperlipidemia, hyperuricemia will be seen in this patient. Pompey's disease, this is actually lysosomal disease, okay, not a completely glycogen. Acid maltase and the alpha glucosidase is the deficiency enzyme. Cardiomegaly, if you see the cardiomegaly, pump, you have the 4 chambers to pump, right? Pompey pump. Cardiomegaly likely to be this patient. Next, Cory's C and D, Cory's debranching enzyme, hypoglycemia and cirrhosis will be seen in this patient. Anderson disease, branching enzyme, most common hepatomegaly with a progressive severe cirrhosis will be seen in this patient. McArdle's, you are going to see this kind of urine in the patients. Cola or the magenta color, and also exercise intolerance will be seen in this patient. Hers disease, only the hepatic phosphorylase, mild hypoglycemia will be seen in this patient. Next, G6PD. This is a X-linked recessive condition. The enzyme is the deficient G6PD. And whenever there is a decreased NADPH, oxidation will be decreased. If the oxidation decreased, RBC is going to die. And anemia, Hen's bodies, here you can see these are the Hen's bodies, okay, and these are the bite cells. It's like a bite, okay, and blister cells. Here you can see this is the blister cells, Hen's bodies. These are the findings that you are going to see on the peripheral blood smear. Whenever they take the fava beans or the malaria drugs, they prone to any other infections, they will be increased hemolysis will be seen in this patient. Next, galactosemia, Galvan-Put gene deficiency, bilateral reversible oil drop cataract. This is the cataract, and bilateral reversible. I am telling this is a reversible. If the patient is stopped on the lactose-free diet, this will be gone. Next, hepatomegaly, absolute breastfeeding is contraindicated. Failure to thrive will be seen in this patient. Next, stop the breastfeeding and lactose-free diet for the 5 years. Benedict test positive and mucic acid test is positive for this patient. Next, coming to the lipids, most important lipids, lecithin, phosphatidylcholine. This is present in the cell membrane and the surfactant. Next, phosphatidylserine. This is present in the apoptosis patient, and phosphatidylinositol present for the second messenger. Next, phosphatidic acid for the cell membrane, which is present, and cardiolipin is present in the inner mitochondrial membrane. Next, fat metabolism. Whenever you take the food, the carbs is converted as a glucose. Excess glucose which will be converted as a DHAP, and further VLDL is going to take that for the fat process. Next, fats directly taken from the food converted as a micelles, which is a minute emulsification substances, and this is taken by the chylomicrons. This is the endogenous process. This is the exogenous process. Next, coming to the beta oxidation, we have to break down those fatty acids. How we are going to break down? This is occurred in the mitochondria. Let's see. First, fatty acid is converted as a acyl coenzyme. Okay. For this acyl coenzyme, it is in the cytoplasm only. Okay. This is converted in the cytoplasm. After this, it is going to converted in the outer mitochondria, between the outer and the inner mitochondria. This is the internal mitochondrial membrane. What happens here? The acyl CoA with the carnitine substances, it is converted as a acyl carnitine with the help of the CAT1 gene. This is the carnitine shuttle. This shuttle is known as a carnitine shuttle. This is the CAT gene. Sorry, CAT enzyme it is going to convert. And within the once it is entering into the mitochondria, this acyl carnitine will be converted as a acyl CoA and carnitine, and both it is going into the one will be going into the TCA cycle and other one will be going into the ketone cycle. Okay, ketone synthesis and the TCA cycle it is going to convert it. This is the beta oxidation known as. Next, alpha oxidation, it is only for the very long chain fatty acids, occurs in the peroxisomes. Omega oxidation, it is only active when the beta oxidation is absent, present in the medium chain fatty acids and occurs in the smooth endoplasmic reticulum. Zellweger disease. What is this Zellweger disease? This is an autosomal recessive PEX gene mutation peroxisomal condition. It can affect the brain, hepato and renal conditions along together. So whoever associated with the Zellweger, this is the appearance of the patient, and they are more prone to increased very long chain fatty acids in the blood and fentanic acid. Next, Refsum disease. This Refsum disease is seen in the patients those who are suffering from the fentanyl CoA oxidase enzyme deficiency, though they are decreased with the alpha oxidation. The symptoms commonly retinitis pigmentosa, ichthyosis and the neuropathy. Next things that you have to take note about the ketone body synthesis. The ketone body synthesis patients will be these occur in the mitochondria, and the rate limiting enzyme is the HMG-CoA synthase. And what are the keto acids involved here? Beta OH or beta hydroxybutyrate and acetoacetate and the acetone. So remember one thing, these are the pure ketones. Okay, so if you test these ketones in the urine, you are going to see this positive. This purple color discoloration when you see in the urine, that is due to Rothera test positive for the ketone body synthesis. Cholesterol synthesis occur in the cytoplasm. HMG-CoA reductase is the enzyme that is the rate limiting enzyme, and same enzyme which will be seen in the statins, right? In the statins, if you block this HMG-CoA reductase, obviously cholesterol synthesis will be stopped. And these derivatives, what are the derivatives that we are going to get from the cholesterol? Bile acids, steroids and vitamin D. Next, lipoproteins. Here you can see this is the structure of the lipoprotein, and these free cholesterol seen completely here. You can see triglycerides and also phospholipids and the cholesterol esterases. Let's see one by one. And here you can see chylomicrons. Chylomicrons is the very largest exogenous, right? Here you can see this is the very largest type, and maximum triglycerides and minimum density. And these are the exogenous type. And what are the apoproteins that is associated? Apo 1, 2, 3 APO124 and B48, C123 and E are the apoproteins that is present. Next, coming to the VLDL, B100, C123 and E. This is the endogenous triglycerides and precursor for the LDL and also IDL. It is going to be the precursor for the LDL and also IDL. Next, coming to the LDL, it is associated with the B100, only B100 only. Maximum cholesterol, very atherogenic condition you are going to see with the LDL. Next, HDL is a very good cholesterol that we had, maximum density and very minimal cholesterol that is present. And what are the present here? APO A123, APO C123 and D and E. And note down that there is no B. Okay, no B is present in the HDL. Ideally will be consisting of the B100 and the ApoE. Next, familial dyslipidemias. In the familial dyslipidemias, here you can see the type 1 is the autosomal recessive. LPL, lipoprotein lipase, is a deficiency. Increased chylomicrons, triglycerides and the cholesterol will be seen in this patient. Hyperchylomicronemia. Here you can see chylomicrons will be increased. These kind of xanthomas will be seen on the patient's skin. Next, type 2 hypercholesterolemia, autosomal dominant, LDL receptor is deficient. So what happens? Cholesterol will be raised and there will be tendon xanthomas, Achilles tendon and this tendon xanthomas, and this condition, the corneal arcus, where you can see the white line. This will be seen in this patient. Next, type 3 autosomal recessive, dis beta lipoproteinemia, ApoE is the deficiency, chylomicrons, VLDL will be increased. Type 4 autosomal dominant, VLDL is only raised, hypertriglyceridemia, so increased triglycerides will be seen. Next, beta lipoproteinemia, chylomicrons, VLDL, LDL will be increased, MTP deficiency. So on the blood smear you are going to see acanthocytes. These kind of acanthocytes you are going to see. Next, Tanger's disease, decreased HDL and it is highly atherogenic. This kind of conditions will be seen. Next, LSDs, lysosomal storage disorders, where there will be Tay-Sachs disease, deficiency of the hexaminodase A, and this is the most common for the cherry red spot in the macula. And Gaucher's disease, overall the most common LSD disease, and there will be, here you can see this kind of like the macrophages is the compelled tissue paper appearances will be seen in the Gaucher's disease. Next, Fabry's disease associated with the alpha-galactosidase A deficiency and angiokeratomas and the acroparesthesias and the cardiac anomalies will be seen in this patient. Next, Krabbe's disease, which will be deficiency of the galactocerebrosidase, and there will be these kind of globoid cells on the peripheral blood smear will be seen, and peripheral neuropathy, developmental regressions and the cranial nerve too will be damaged in this Krabbe's disease. Next, Niemann-Pick disease, which will be deficiency of the sphingomyelinase and the sphingomyelin, and hepatomegaly, cherry red spots, which will be common. Cherry red spots commonly with the Tay-Sachs, then with the Niemann-Pick and the Gaucher's. Next, purines will be, purine ring will be formed by the glycine, aspartate and the glutamine, and this will be having the two pathways, de novo synthesis and the salvage pathway. De novo synthesis is seen in the every structure except brain, RBC and the WBC. For the brain, RBC, WBC, there will be salvage pathway. And rate limiting enzyme for the de novo synthesis will be PRPP glutamyl amidotransferase. And the conditions that is associated with the purines metabolism, Leishman syndrome and the Kelly-Segmiller syndrome. So Leishman syndrome is the X-linked recessive HGPRT deficiency. Here you can see this is the condition of the Leishman syndrome. There will be self-mutilation, mental retardation, gouty arthritis, because HGPRT is going to block the xanthine oxidase and there is no process there, so there will be increased uric acid will be seen. Kelly-Segmiller is associated with the same Leishman, but it is a very partial defect. Next, pyrimidines, which will be aspartic acid, glutamine and rate limiting enzyme for the CPS, the carbamyl phosphate synthase 2, and the site is mitochondria and cytoplasm. The only disease that is associated here is the hereditary orotic aciduria, and the enzyme that is deficiency is UMP Synthase is a deficiency, mental retardation, microcytic anemia. If they are not responsive for the B12, B9 medications, so likely to be the hereditary orotic aciduria.